right, here we go with section 17.4. We're going to tackle translation. So it's really a great title to this section. It is an RNA-directed synthesis. You're going to see all three forms of RNA used to actually read the messenger RNA and build a protein. So in the first section, they talked about the molecular components of translation. And here's where they get into the details of transfer RNA. So a tRNA is really one long RNA molecule. And so if we take a look on the next page, which they reference, you see, what? All right, welcome to section 17.4. We finally get to translation. The title of this section is very appropriate. The whole way to make a polypeptide or a chain of amino acids is truly RNA directed because you're going to see all three forms of RNA used to do this. They begin by talking about the components, which makes sense. So we have the first component, transfer RNA, which is what brings in the amino acids. It is actually made of a long strand of messenger RNA, and they have some nice pictures here on the next page that actually show, the transfer RNA. So it looks like a clover leaf, and it's one strand. Notice it's hydrogen bonded in certain locations to give that shape. And you can see down here, the three codons, that complementary base pair with the codons on messenger RNA. And that's just one way in which the cell knows we're bringing in the right amino acid. And notice that attachment site is right up here. The other way it knows is by actually making sure it bonds the right amino acid with, we'll say, an empty tRNA or an inactive tRNA. So the purpose is to bring an amino acid. Well, it's not bonded here. Its own tRNAs bring specific amino acids, and so we have enzymes that are talked about on the next page, called amino acid tRNA synthetases that actually help bond the right amino acid to the correct transfer RNA. So, sorry, and I forgot to focus here. You can see other forms of the transfer RNA kind of shown in a more ribbon format, and then in most of their drawings, they keep it simplified, just noticing the attachment site for the amino acid and then the anticodon. Now, if you recall, there are 20 different amino acids that are used to make polypeptides, and polypeptides are hundreds to potentially thousands of amino acids long, so it's just an arrangement of them, but of course in a specific order, just like the alphabet is used in a specific order to make words and sentences to make sense. So how do we know we're bringing the right amino acid? So you basically have to put the right amino acid on the right tRNA, and that's highlighted through the enzyme amino acid tRNA synthetases here at the top of the next page. So I believe you were given this picture to label in your notes. So make sure you take time to go through this figure and do just that. Really briefly, so notice we have a tRNA here, It doesn't have an amino acid attached to it. Which one of the 20 should it bond to? Well, notice this enzyme right here, which is really big, has multiple locations for things to bind in its active site. And the first one, notice, it's going to be specific to the anticodon. The other place it's going to be specific is to what amino acid. And then enzyme is needed, in order to bind this. So you see the use of ATP here. And the enzyme will then bond the right amino acid. And then now this tRNA molecule will be in the cytoplasm ready to help build a protein. So here's a fun fact for you. So they talk about it over here, and then they continue on to the next page, finishing up that description of figure 17.6. One of the big things here, as you can see with this sticky note, as you read through, there's something called a wobble. And you definitely need to familiarize yourself with that. So the rules for base pairing is not always like all three codons have to match with all three anticodons. The first two are very strict, as you can see in my sticky note here. The third one may vary, and they talk about the A and G kind of wobbling. And that's how you can have 64 different codon codes, but only 20 different amino acids. And you'll notice in the chart, it's that third letter that can vary that still brings the same amino acid. Next, I'm going to read the sticky note, they start talking about what's going on in the ribosome. Well, the ribosome was actually made up of RNA and some proteins that hold it together. And the ribosome itself, and you can see in this picture right here, is actually made of two pieces. You have a small subunit and a large subunit. And the one reason why I'm not the biggest fan of this picture is because technically these are never together unless they're involved in translation. So this right now is not involved in translation. These should be separated. Kind of like a hamburger one, that's how I think of it, right? There's a top and a bottom, and the only reason they come together is when there's your burger or whatever it is you eat, okay, in the middle is where they come together. Otherwise, they can separate at any time. Moving on, okay, we continue reading about the ribosome, and they go into some detail here again about like prokaryotes and eukaryote differences. Don't worry so much about it, but there is actually something here I would know. Oh, so first, my sticky note here, important, make sure you note, right, again, those large and small subunits do not come together unless the messenger RNA is attached and we're beginning translation. So if there's no mRNA, the large and the small, like if they're my hands, they're going to be floating around separately. Until mRNA attaches, so let's say that's pencil, then a large subunit will come in and then read the messenger RNA. Going back, one big thing to know with the differences between prokaryote and eukaryote that I would take notes on is the ribosomes. Okay, they're a little different in size between a prokaryote and eukaryote, and medicine has taken advantage of this by developing some antibiotics that are able to only tackle prokaryotic ribosomes and not eukaryotic ribosomes. Next, they start talking about the different locations as you saw in E, P, and A. You need to know, okay, all three of those sites, the E, P, and A locations of the ribosome. I will let you read through them. It's pretty straightforward. All right, how do we actually make that polypeptide? So if we, making our polypeptide, so as you can see, as I talked about, right, those subunits are separated from one another. What happens is the messenger RNA, okay, attaches to the small subunit. And if you notice right up here, it's the same stages as we had for transcription: initiation, elongation, and termination. So if we look at this picture, which I believe you have unlabeled, make sure to label it. And if you notice, there's always a start codon. People, yes, you have to know what the start codon is, and they discuss it in this paragraph up above. It is A, U, G, and eukaryotes. Don't worry about prokaryotes. Always you need to know the start codon, and they even highlighted here on messenger RNAs, AUG. And that means it's always the same tRNA with the same amino acid. For short, it's MET. You need to know that. It's short for methionine, but as long as you remember MET or MET as the amino acid, you're in a great place. Once those bind, so messenger RNA binds to the small subunit of the ribosome, tRNA with the right complementary anticodon comes in, and that is when the large subunit can attach. So we have now initiated translation. Then in the next page, they talk about elongation in this big old picture. I'll be very brief with it. You need to read your details. So if you take a look, you've got your A, P, and E site. The polypeptide chain, okay, is attached to the last tRNA molecule that was red, and that is in the P site. So the A is now open, and the codon, okay, in messenger RNA, the 3 nucleotide bases are sitting there. So the next next tRNA molecule comes in. Now there are actually lots of them floating around here. They're not showing that. So you might have a couple try to come in, but if they don't complementary base pair correctly, codon and anticodon, it's going to get booted out. So using some energy, okay, we have a right fit. Okay, we're going to, with the amino acid that is in the A site, move over our polypeptide chain to make that amino acid next in the chain. So if you notice, right, this tRNA had just come in with its amino acid. The polypeptide gets moved over, okay, and bonded here with some more energy. And then the ribosome shifts. So if you notice, now my polypeptide chain that was in the A site is now in the P. And this doesn't have anything on anymore, so it gets booted out. This tRNA is, I would say, inactive because it doesn't have an amino acid. So it'll find the enzyme again that can help find the right amino acid, and voila. To finish this process, termination, okay, you have your nice pictures up here. And basically there are stop codons. These do not code for amino acids. Okay, so if you take a look here, there's this codon here. You don't need to memorize the 3 ones. They'll have them for you. What this actually codes for is a protein called a release factor. So if a tRNA comes in here, it will not be able to bond. Only this protein will. When it bonds in the A site with the codon for stop, that basically tells the ribosome to release everything. So notice this polypeptide chain gets broken off, and here it is. Then you have your tRNA that doesn't have an amino acid. It goes and finds one, and the ribosome dissociates, and the messenger RNA is left to float around. There you go. We will start our next video with completing and targeting functional protein.